Okay, so this is actually the last of the protein function videos. We're actually going to be talking about fetal hemoglobin, which we were picking up from the last video. And I'm going to remind you that the fetus has to have gas exchange across the placenta because there's no connection between the circulatory system of the fetus and the mother. So it's the difference in the hemoglobins that accounts for the ability of the fetus to get oxygen from its mother. Okay, so the gas exchange is happening in the tissues, in the placenta, where most hemoglobin likes to let go of oxygen, but placental hemoglobin or fetal hemoglobin, HbF, is actually going to have a higher affinity for oxygen than the adult hemoglobin. Because remember, I mentioned in the last video that the fetal hemoglobin replaces the beta chains with gamma chains. That's gamma, right? Gamma is the Greek letter, the third letter of the Greek alphabet. Okay, so it has an alpha 2 gamma 2 structure instead of the alpha 2 beta 2 structure of adult hemoglobin. Why does fetal hemoglobin bind oxygen more tightly than adult hemoglobin? Because its gamma chains have serines instead of histidines that line that core, and so they're missing 2 of the 8 positive charges that I mentioned before that stabilize the BPG binding. So with less of the hemoglobin, the fetal hemoglobin bound to BPG, more of it is in the R state and can capture the oxygen. And so when the oxygen is being transferred to the tissue from the adult hemoglobin, it can be picked up by the fetal hemoglobin because it has that higher affinity for oxygen. Now keep that in mind, because as we move over to a molecular disease of hemoglobin, now we're talking about adult hemoglobin, sickle cell anemia, you're going to see how fetal hemoglobin plays into this story as well. So sickle cell anemia patients have sickled red blood cells, and this causes a lot of problems with the ability to transport oxygen, because the red blood cells are chock full of hemoglobin. That is the hemoglobin delivery system. It's the red blood cells. So if they're misshapen, they can actually get stuck in the capillaries, or more likely filtered by the spleen. And that means that most of your hemoglobin is not making it to the tissues, and that's where the anemia comes in. And so it also can lead to a lot of pain in the bones and in the muscles, and it can even cause death. Okay, so what's happening in sickle cell anemia is that the cells, the red blood cells, sickle because of a single mutation, a single amino acid substitution in the beta subunit. Once again, we're talking about the beta subunit of hemoglobin, adult hemoglobin, that changes a glutamic acid at position 6 of the beta chain to a valine. So that's a big change. That's an acidic amino acid changing over to a non-polar amino acid with a hydrophobic side chain. And that hydrophobic side chain is actually exactly the right hydrophobicity and shape to bind into another beta chain on another hemoglobin molecule, and you form these non-covalent polymers of hemoglobin molecules that form rods, very stiff fibers inside the red blood cell, and they poke the membranes of the red blood cell, and that's what gives them that sickle shape, that shape like a pinched red blood cell. So it's the specific aggregation into non-covalent polymers that poke the erythrocyte membrane into that sickle shape. And remember, it's a single glutamic acid to valine substitution that's actually affected at the DNA level that leads to this substitution, of course, at the protein level, which leads to sickle cell anemia. These are healthy red blood cells, They have that donut-like shape, and their job really is to just transport around a whole lot of hemoglobin to your tissues. This is what a sickle cell looks like here, and it's pointy like this because if you open it up, you see fibers made up of hemoglobin. So these are hemoglobin tetramers, and what you can see is the beta subunit on one tetramer is interacting with the beta subunit on another tetramer. So there are two positions on different beta subunits that are interacting to bury that new valine, used to be glutamic acid. Okay, so we're going to have another look at that. Close up, we're looking at a phenylalanine that lines a pocket on one beta subunit that interacts, again, in the T state only of the beta chain of another subunit, the valine. Remember, there are two beta chains in every hemoglobin. So one of the beta chains, phenylalanine, is here, and the other beta chain, beta 1 and beta 2, interacts with that pocket just perfectly. So the hydrophobic pocket fits the valine shape, but also buries hydrophobicity against hydrophobicity, freeing up water by the hydrophobic effect. Okay, so let's zoom out again, and let's have a look at beta 1 and beta 2 to help us understand how the beta subunits can fit with that valine side chain into a hydrophobic pocket of another tetramer. Okay, so forming these polymers, and again, these are non-covalent polymers, arises because the valine is replacing a glutamic acid. Glutamic acid would not be happy in this hydrophobic pocket because there's a huge thermodynamic penalty burying a charge, a full charge, never mind a partial charge, into a hydrophobic space. So glutamic acid at this position would prevent this type of interaction. But a valine at that position actually helps the interaction. All right, so let's have a look at oxyhemoglobin. Oxyhemoglobin is smooth. No block extending from one of the beta subunits, and no pockets either. Okay, so this is oxyhemoglobin. Beta 1, beta 2, alpha 1, alpha 2. Deoxyhemoglobin A, this is, again, is the healthy adult hemoglobin, has a pocket in the beta 1 chain. Okay. So on this side, on this face, there's also a pocket in the beta 2 chain, but it's into the board. Okay. And if you look at oxyhemoglobin S, this is the sickle cell, this is the mutation. You have that valine protruding from this side of the beta chain, and another one protruding on the other side of this beta 1 chain, because these are both beta chains. Okay, and so now you put deoxy, this is a T state, right? The T state, this is the R state, T state, R state, T state. Oxy hemoglobin S does not form these polymers, these chains that sickle the cells. But deoxy hemoglobin S has the block extended, that's the valine, and it has the hydrophobic pocket on the front here, so that now they can interlock, and you actually get those polymers forming only in the deoxy state, the T state of sickle cell hemoglobin. And they fit together like this, right? If you're looking at this face, you see beta 1 has a pocket because you're in the deoxy state, and beta 2 has a valine sticking out in both the oxy and the deoxy state, but just so happens to fit beautifully into the pocket that's available only in the deoxy state on this side of beta 1. And that's how you get these long polymers, non-covalent polymers, It's the hydrophobic effect that's driving the polymerization of hemoglobin that sickles the cells and causes sickle cell anemia. And when you look at them under the electron microscope, this is what you see. This is a sickled cell, and it's broken open, and these fibers that you see protruding out of it are hemoglobin S. In the deoxy state, they get trapped in these polymers, and they form this elongated sickle structure that gets trapped up by the spleen, which lowers the amount of oxygen getting to the tissues of these adults and causes sickle cell crisis. Now, how can patients who are undergoing sickle cell crisis be treated? Well, it turns out that hydroxyurea is a treatment for sickle cell anemia, and it was known to help, but it really wasn't understood how it helped until molecular biology came along. And what we learned is that hydroxyurea stimulates the production of hemoglobin F. Hemoglobin F gene is turned off typically, but if you treat with hydroxyurea, you activate expression of hemoglobin F, fetal hemoglobin, which does not have the beta subunit and therefore does not have a pocket or a protrusion, because there's now that valine mutation is not there. Instead, it has gamma subunits and therefore will not sickle the cells. So that's how hydroxyurea helps treat sickle cell crisis. Now, where did this mutation come from, and why is it so prevalent? It came from the disease malaria, which is caused by the parasite that causes malaria, which is prevalent in tropical climates. And so you'll see that in Southeast Asia and in Africa, you have a lot of overlap between malaria and sickle cell disease. And the sickle cell gene actually confers on the patients who are heterozygous. So they have one copy of the sickle cell gene and one copy of the wild type gene. So they have fewer crises, but they actually survive malaria because the malaria parasite infects the red blood cells and will be filtered out by the spleen and allow these families that have this inherited trait to survive. So the sickle cell trait was actually selected for in order to protect the population from malaria. Now, if both parents have the sickle cell trait and you have a child with sickle cell disease, then they're more likely to die and not benefit the population. But having this heterozygous trait actually does confer resistance to the population, resistance to malaria. And this is the malaria parasite in one of its life cycles right here inside a red blood cell. It's transmitted by mosquitoes. There are other mutations that alter hemoglobin's function, and as one of the first proteins ever to be studied at the structural and molecular level, we've been able to come up with many more than are shown in this slide, but this is taken from your textbook, and it shows you the actual molecular problem with each of these thalassemias and other mutations that cause different types of anemias. All right, so chapter 7, we covered 2 different types of protein function. There are as many different protein functions as there are proteins, so we couldn't cover everything. There are some more examples in chapter 7 that you may want to study if you're going on to take the MCATs. But what we covered here and what I'll expect you to know for the exam are the hemagglutinin coiled coil used for viral proteins to pierce and pull your membrane into their membrane so that they can replicate and go on infecting people. And hemoglobin and myoglobin. This one is from chapter 7 of the Vogt and Pratt. Myoglobin is, again, this is summarizing what we just learned, is an oxygen storage protein. There is no regulation. It's a monomer. It doesn't have cooperativity. It doesn't have allosterism. Hemoglobin is for oxygen transport, so it has to let go of it, and it's helped along by allosteric regulators, carbon dioxide and protons. These are connected by the Bohr effect and the 2,3-BPG that we talked about. Fetal hemoglobin has the gamma subunit and not the beta subunits, and so it doesn't bind to BPG very well, and therefore it has tighter binding to oxygen, higher affinity for oxygen, and it's able to pick up the oxygen across the placenta through gas transfer. Sickle cell anemia is a molecular disease of hemoglobin due to a glutamate to valine mutation in the beta subunit that leads to the aggregation of hemoglobin in the T state, deoxyhemoglobin. It forms polymers, causing sickling of red blood cells and anemia. And the trait was selected for survival to the malaria parasite.